Have you ever wondered what makes you, you? For a long time, we thought the answer was simple: our genes. The idea was that our DNA is like a detailed blueprint, dictating everything from our eye color to our personality, and maybe even who we are attracted to. But what if the blueprint isn't the whole story? What if there's another layer of information that tells our body how to read that blueprint? This is where the fascinating field of epigenetics comes in. And it's changing how we understand ourselves, right down to the core of our identity. To really grasp this, let's talk about genetics versus epigenetics. Think of your DNA as a massive cookbook, filled with thousands of recipes. This cookbook is your genetic code. It contains all the potential dishes your body could ever make. Genetics is the study of the cookbook itself, the sequence of letters in the recipes. If you have a recipe for chocolate cake, genetics is about the specific ingredients and instructions written on that page. It's the fundamental code passed down from your parents. Epigenetics, on the other hand, isn't about the recipes themselves. It's about how those recipes are used. Imagine someone went through your cookbook with sticky notes and paper clips. A sticky note might say, "Make this recipe more often." Or a paperclip might hold 2 pages together, making one recipe impossible to read. These epigenetic marks don't change the actual recipe. The DNA sequence remains the same. But they do change which recipes are read and how often. They're chemical tags that attach to our DNA, acting like switches that turn genes on or off, like volume knobs that turn their activity up or down. So, while your genes are the hardware, your epigenome is the software that tells the hardware what to do. This is a crucial distinction. It means that 2 people can have the exact same DNA, but express those genes in very different ways. And where do these epigenetic changes come from? They can be influenced by a huge range of factors throughout our lives. Everything from the food we eat, to the air we breathe, to our stress levels and social experiences can leave these little marks on our DNA, shaping how our genes behave. This brings us to a really powerful way to see this in action: twin studies. Identical twins are nature's perfect experiment. They develop from a single fertilized egg that splits in 2, which means they start life with virtually identical DNA. They have the same genetic cookbook. If a trait were purely determined by genetics, if it were just about the recipes in the book, then identical twins should be identical for that trait 100% of the time. If one twin has it, the other one must have it too. This is called a concordance rate. For purely genetic traits like eye color, the concordance rate is extremely high. If one identical twin has blue eyes, the other is almost certain to have blue eyes as well. This tells us that eye color is heavily, if not entirely, dictated by the genes themselves. But what about more complex traits, like aspects of our identity and behavior? What about sexual orientation? If being gay, straight, or bisexual is written directly and unchangeably into our DNA, then if one identical twin is gay, the other one should always be gay too, right? The concordance rate should be 100%. For decades, scientists have been studying this very question. They've looked at thousands of pairs of identical twins to see how often they share the same sexual orientation. The results are incredibly revealing, and they might not be what you expect. Study after study has found that the concordance rate for sexual orientation in identical twins is not 100%. It's not even close. While there is a higher chance for an identical twin of a gay person to also be gay compared to the general population, it is far from a certainty. The numbers vary between studies, but they consistently fall somewhere between 20% and 50%. Let's break that down. This means that if you take a group of identical twins where one twin identifies as gay, in about half or even more of those cases, the other twin with the exact same DNA does not. This single piece of data is profound. It's a powerful indicator that while genes clearly play a role, there is a genetic component. Which is why the rate is higher than in fraternal twins or siblings. They are not the whole story. There has to be something else at play. If it's not just the genes, then what is it? This is where we circle back to epigenetics. The sticky notes and paper clips on the DNA, scientists now theorize that epigenetic markers could be a key part of the puzzle. Perhaps specific epigenetic patterns developed very early in life, maybe even in the womb, influence the pathways that shape sexual orientation. 2 twins could have the same genes, but different epigenetic modifications to those genes could lead them down different paths. Think about the environment in the womb. Even identical twins can have slightly different experiences. One might have a slightly better position or a more favorable connection to the placenta, leading to minor differences in hormonal exposure or nutrient supply. These subtle differences could be enough to create different epigenetic signatures on the genes related to brain development and sexual differentiation. One twin's volume knob for a certain gene might be turned up, while the other's is turned down, all because of tiny variations in their earliest environment. This epigenetic model helps explain why one twin might be gay and the other straight. Despite sharing the same genetic blueprint, it bridges the gap between the nature of our genes and the nurture of our experiences, suggesting they are deeply intertwined. It also helps to explain the beautiful diversity of human sexuality. It's not a simple on-off switch located on a single gay gene. Instead, it's likely a complex interplay of multiple genes and the epigenetic factors that regulate them, creating a spectrum of attractions and identities. This understanding moves us away from a rigid, deterministic view of who we are. It shows that our biology is more like a dynamic, responsive system than a fixed set of instructions. Our genes provide a starting point, a range of possibilities, but our epigenome helps to shape the final outcome. This doesn't mean orientation is a choice. Epigenetic changes that influence something as fundamental as attraction are thought to happen very early, long before we have any conscious control. They are as much a part of our biological makeup as the genes themselves. So, when we look at identical twins, we're not just seeing two people who look alike. We're seeing a living demonstration of the intricate dance between genetics and epigenetics. They show us that identity is not pre-written with indelible ink, but rather sketched out with layers of complexity added throughout our development. The data from twin studies is a powerful challenge to simplistic explanations. It tells us that the answer to what makes us who we are is not a simple one. It's a combination of the genetic book we are given and the unique way that book is read throughout our lives. The world of epigenetics is still a new frontier, and there's so much more to learn. But it offers a more nuanced and accurate picture of human diversity, celebrating the complexity that makes every single one of us, even identical twins, completely unique. If you found this exploration into the science of identity interesting and want to learn more about the complexities of human sexuality, be sure to check out my channel, Sex Education Repo. We dive deep into these topics to bring you a clearer understanding.